Can you guys still see it? I have on present mode.
Yes,
Yes.
we can see it.
Thank you. So my name is Abigail Fenzik. I'll be presenting on Ideonella seconensis. It's a plastic eating bacteria that was recently discovered in Japan. They discovered it living inside of a plastic water bottle in a landfill or a recycling plant. It was a recycling plant, I'm sorry. So it's still being researched and genetically modified worldwide in order to, the goal is to strengthen the plastic eating properties, which are the enzymes that break down the plastic bonds. I have 2 videos that I'm going to be showing them in parts throughout my presentation. They're both interviews of a scientist, John McGeehan. He is a professor at Portsmouth University in the United Kingdom. So they're 2 different videos, but they're both interviewing the same scientist.
It's estimated more than 8 billion tons of plastic has been produced since the 1950s. In 2016, a team of Japanese scientists made a discovery that had the potential to turn the tide in the fight against plastic.
They were scraping around in the soil and wastewater runoff of a recycling facility in Japan. They actually found a bacteria that remarkably was living off the plastic bottles themselves. So they took that back to the lab and they were able to culture it, just feeding it a diet of plastic, PET plastic. That's the plastic for bottles and clothing and carpets and that sort of thing. So it's a very common plastic in the environment. They've evolved to eat various types of food sources, and what seems to be happening in that rubbish dump in Japan is that they're secreting a couple of enzymes, and those enzymes actually break apart the polymer chain in the plastic, providing molecules as food.
So that was the first part of that video. And then I have a small part of this video as well.
The world's plastics problem is out of control. If it keeps piling up, plastics in the ocean could outweigh fish by 2050. Plastic is difficult and sometimes impossible to recycle, and it doesn't break down naturally. But a team of scientists have stumbled on a discovery that could change the way we deal with plastic forever.
So this is the biochemistry lab, and this is where everyone is working. So we followed a Japanese study that came out about 2 years ago, where they made this startling discovery of a bacteria that lives off plastic in a recycling dump. Essentially, this bacteria is evolved to eat plastic, and it produces an enzyme that breaks down plastic into its original components. So that's really where our story began. We wanted to find out how this enzyme worked. And by just fiddling about with the DNA sequence, we accidentally made a more efficient enzyme that's about 20% faster than the natural enzyme. Science is a bit like that. You do the experiment, you can predict what might happen, but often you're wrong.
And I know it was a little bit repetitive for the beginning of that, but I really liked how he put it in the end, where with science, sometimes it's almost an accident how things are discovered. And also, it's very important that they have been working to make it more and more effective. And this could become the new way that we recycle throughout the planet. So the process of breaking down plastic starts with obviously the plastic, which is composed of ethylene glycol and terephthalate. I'm so sorry. Terephthalic acid brought together by
pH is silent. Yeah, I mean the pH is silent in that, so it's terephthalic acid or terphthalic acid.
Thank you so much. terephthalic acid brought together by polymerization. The ethanol glycol has hydroxyl group and the terephthalic acid has a carboxyl group. These bond together through ester bonds, and they are then bonded together more to create the polyester chain that was also mentioned in a video I'm going to show. That will also have the diagrams, so it'll be a lot more clear. The first enzyme, because there's actually two enzymes in the process of breaking down plastic in this bacteria. The first enzyme is called petase. It breaks down the polymer chain, leaving what they call MHET, or mono 2-hydroxyl-ethyl terephthalate. This is using the process of hydrolysis. The second enzyme, MHETase, also uses hydrolysis to break the bond between, the bonds within MHET, leaving ethanol glycol and terephthalic acid that can be polymerized into new PET plastic, or the ethanol glycol could be used as a fuel and there's a lot of other applications as well. And I will show the rest of this video.
Professor John McGeehan and his team at Portsmouth University in the UK are one of several groups around the world now investigating the potential of plastic-eating bacteria.
So to understand the process of how this works, you kind of need to understand what a polymer is. So PET plastic is an incredibly common plastic in the environment, and it's made of two building blocks, one called ethylene glycol and the other called terephthalic acid. And basically these building blocks are kind of held together by bonds called ester bonds. So then you get poly, lots of ester, polyester, and this is our plastic. So if you look on your clothing, you'll probably see a label saying polyester. That's all it is those building blocks linked together in long chains that create these plastics. And all an enzyme needs to do is come along and snip those bonds like a big pair of molecular scissors, and then you get the building blocks back again.
So you found a bacteria that's living in a rubbish dump. How do you then get enzymes out?
Actually, these days it's very simple. You grow the bacteria in the lab, and then coding within that bacteria is DNA. It's like a 4-letter code that codes for the instruction manual for that enzyme. We can pull that out and make it synthetically in the lab. These are the two individual enzymes that are produced by that bacteria in the rubbish dump found in Japan. And what we did in the laboratory is take those two separate enzymes. The first one is called petase and the second one's called MHETase. And we physically linked them together to make this chimeric enzyme. And we found that this one was around 6 times faster than the original natural enzyme that we originally studied. So what we can show here is that we can take some learnings from nature, take them back to the lab, and then adapt things to make them faster and more efficient, which is what we need if we're going to have a economically viable recycling solution. Enzymes are really ubiquitous in nature, and they're used for making things and breaking things. So the human body is full of enzymes. We use enzymes every day in our biological washing powders. They contain those powdered enzymes that can break down grass stains and our food stains. So we're actually, as a technology, it's very mature. So then turning enzymes towards problem materials like plastics really seems like a sensible way forward. The sorts of enzymes we're using today in the food industry and the pharmaceutical industry, they were also very slow when they were first discovered. And over years of generations, we've managed to improve them to be, in some cases, 100 or even 1,000 times faster than the original natural variants. So this gives us hope.
All right, and then to conclude,
Professor John McGeehan.
This could be the possible future of recycling, like how enzymes revolutionized medicine, food, and other industries. Soon, enzymes will also be helping save the planet by creating a new way to handle recycling plastic. Although PET plastic is only part of the high carbon emissions human create, scientists may be able to apply this technology to other forms of waste as well. And this video isn't going to talk about it, but I just really like how this scientist talks about this project. So I'm going to just share it. It's about a minute.
So what we can see at the moment, if you take a plastic bottle, a fizzy drinks bottle, and you put it in a test tube with some of our enzyme, it'll start digesting away the surface over a couple of days.
Seeing if we can identify some of our protein that we've expressed through our mammalian cell lines. So
Excellent. If we can do this with a fast enough enzyme on a bigger scale, you can imagine collecting plastic, and what would happen is the solution would go clear as the building blocks were broken down. So you'd have this clear solution of chemicals that you can then remake plastic with. I think there's been a huge amount of doom and gloom stories around plastics, and justifiably, because it's a terrible environmental scourge. But this is a story where we've got some hope that we can actually put together. And my email box has now got 500 unread emails with companies saying, can we work with you? Can we help? Certainly since this story came out, I could count the number of hours I've slept on one hand.
All right. So that is the end of my presentation. If anyone has questions.